Hi everyone, we're Group 25 and today we'll be presenting on Hutchinson-Gilford-Progeria syndrome. HGPS is an autosomal dominant genetic disorder that causes rapid aging in children. The average lifespan for individuals with HGPS is about 13 years, and symptoms include growth failure, loss of hair, loss of body fat, and joint stiffness. This disease is caused by mutations in the elamin A gene, which encodes for lamin A, a structural protein that provides stability for the nuclear envelope in cells. Today we'll be going over one of these mutations and its effects on nuclear assembly and organization.
Lamin proteins are intermediate filament proteins that provide structure for animal cell nucleases. Two main types are lamin A and B. Lamin B is expressed in most cells, while lamin A is only expressed in differentiated muscle cells after birth, which is why progeria is so difficult to diagnose because it's only seen postnatally. And some key functions of lamin A is that it's involved in organization of chromatin and nuclear membrane structure.
The elamin gene is 3,259 base pairs long, and the transcribed pre-mRNA undergoes alternative splicing into mRNA for lamin A and C proteins. The healthy lamin A protein is 572 amino acids long and requires extensive post-translational modification on the carboxyl terminus that occurs in a multi-step process to become fully mature. The protein is comprised of an amino terminal head region, central alpha helical regions separated by linkers, and a 9-strand beta sheet region resembling an immunoglobulin fold before the carboxyl terminal tail.
So we decided to look at a paper from Timon et al. 2009, which was aimed to understand how lamins form into higher order structures and how the E145K mutation impacts nuclear architecture and chromatin organization. Previous studies found that the LA delta 50 mutation is correlated with the loss of heterochromatin in epigenetic marks, so they asked if this also applies to the E145K mutation. So for figure 1, they looked at lamin alterations in the E145K nuclei. They stained E145K nuclei in control fibroblasts with anti-LAC, anti-LB1, and HOCUS, which stands for DNA. They found that LAC is mainly associated with the outer region of the lobules, which stain weakly for LB1 and surround a euchromatic region, which is explained by the limited HOCUS staining in that region. However, B-type lamins were stained mostly by proximal regions of the lobules, and associated mainly with heterochromatin. And you can see that there's severely lobulated nuclei, while normal fibroblasts were free of lobules. In part B, they did an immunoblotting analysis of these cells, and they found that there's a lesser extent of LB1 and LB2 in comparison to controls, and LA and LAC are expressed at levels comparable to controls. So in figure 2, they focused on mislocalization of heterochromatin, centromeres, and telomeres. So as a background, in interphase, gene-rich chromosomes are likely to be internally located, while gene-poor chromosomes are likely near the periphery. Centromeres are situated near the nuclear periphery as well, and telomeres should be localized throughout the entire cell. So in part A, for heterochromatin localization, they did a staining of H3K9-methyl-3 and H3K27-methyl-3, and they found that H3K9-methyl-3 is in the central region of the nucleus in mutant cells, while it's enriched in the nuclear periphery in controls. And they found that the H3K27-methyl-3 is distributed equally throughout the bulk of the cells of the nucleus. For centromere localization, the mutants in controls were stained with anti-LA crest antiserum for centromeres and HOCUS, and they found that the centromeres were clustered in the central region of the nucleus in the mutant cells, however, through the entire periphery in the control cells. And lastly, in part C, they looked at telomeres, and they found that many telomeres were not interspersed with centromeres and also were likely associated with the lamina.
To conclude, the paper showed that there was a reduction in peripheral heterochromatin compared to the controls. The LA delta 50 protein phenotype is likely from abnormal accumulations of lamin at the nuclear envelope, while the E145K phenotype is from abnormal assembly and polymerization of lamin A. Both the E145K mutation and LA delta 50 protein expressing cells result in downregulation of B-type lamins and a premature rest of cell cycle. And it's known that expression of LA delta 50 protein in mesenchymal adult stem cells causes changes in the differentiation pathway. So the next step would be to see if the E145 mutation can yield a similar result.